mynews.studio.
Picture this. In a Beijing lab, rice plants are glowing under ultraviolet light. Each leaf holds a genetic signature that could transform farming and medicine. But here's the real pivot. What happens when a breakthrough allows us to edit entire chromosomes, leaving no trace behind?
The date today is 2025-086. Let's get right into it. Chinese researchers have managed what used to be considered impossible, editing entire chromosomes in plants and animals with total precision. They're calling this programmable chromosome editing, or PCE. So how does this actually differ from CRISPR-Cas9?
Think of CRISPR-Cas9 like a molecular scalpel. It's excellent for small, targeted edits, but not for sweeping changes. PCE is different. It combines three innovations: asymmetric recombination sites, AI-guided protein design, and a prime editing method. With this trio, researchers can insert, delete, swap, or even flip massive stretches of DNA, sometimes whole chromosomes, without leaving any genetic footprints.
No genetic footprints at all? That's new. And they used rice as the first test, correct? What did the experiments actually involve?
Right, rice was the model. The team performed several kinds of edits. They inserted a fragment nearly 19,000 base pairs long, replaced a 5,000 base pair segment, inverted a region that was 12 million base pairs, deleted 4 million base pairs, and even relocated entire chromosomes. The result? Rice plants with stronger resistance to herbicides, and importantly, no leftover signs of editing.
That's a substantial step forward. But how do they make sure it's safe? What checks are in place before this can be used in fields or clinics?
For crops, the process starts with molecular characterization to rule out unintended changes. Then come multi-generational studies to check if the traits stay stable, plus environmental assessments. If we talk about humans, next steps are animal models, followed by phased clinical trials. These look for off-target effects, immune responses, long-term impacts. Regulators will also require data on allergenicity, toxicity, and, if it's food, nutritional equivalence. Even after approval, post-market surveillance keeps watch for delayed effects.
So realistically, we're looking at years before this reaches dinner plates or medical clinics. But if it does work, how could this reshape agriculture?
It could be transformative. Imagine crops engineered to resist pests, drought, and disease, customized with precision and speed. With climate change and population growth pressuring the food supply, this approach could play a major role in food security.
And in medicine, does this open the door to correcting large-scale genetic disorders?
That's the hope. By addressing chromosomal abnormalities directly, there's potential to treat some genetic conditions at their root without adding foreign DNA or leaving scars that could cause complications down the line. But safety and ethics have to lead the way.
Let's touch on ethics for a moment. How are regulators